This is Shelley Craft. We're coming to you live on location at the William E. Hanna Jr. Innovation Center at Shady Grove in lovely Rockville, Maryland. I have with me Dr. Martha Knight from CC Biotech LLC, a private company. Welcome to SNNLive.
Thank you very much. I'm happy to be here.
It's good to have you. Now let's get right into what CC Biotech does.
Okay, maybe literally, CC in the name means countercurrent, or really centrifugal chromatography. What we're developing is a new way of purifying molecules, and this will have effect in the analysis of molecules, like in proteomics, the analysis of proteins, also in the scale-up in the biotech industry, where they're making large amounts of protein-expressed therapeutics, and this would be a very seamless method of purifying them in a flow-through method.
How are they done now?
The present technology usually uses a column of packed material that can affect separation of the molecules. The packing material or solid supports are very expensive to make, and in many cases, especially in the biotech industry, they will be disposed of after an initial use. Well, this new technology is simply a flow-through system of tubing going through centrifuge coils, and the equilibration or partitioning of the molecules between two liquid phases, no expensive solid support. The molecules can be very effectively purified and collected out the other end.
Do you have a sample anywhere around?
Well, actually, I have the frame, since you asked, of the first rotors that we had built, and I'll show you the simple format of it is right here. This is a rotor that can be placed in a centrifuge, and in the channels of this rotor we have tubing wound around in loops going in and out. So the sample comes in and travels through these loops and out the other end. In the centrifugation, there's a centrifugal force field, and there's the phases or mixing the phases of the liquid phases, and the molecules get separated coming out the other end. Now this is usually filled with plastic tubing, and then we can use solvents to go through it, and we have a cap on top of this, and then this is placed in a centrifuge.
So does it take less time?
It may take equivalent amount of time compared to some purification processes, like larger scale high-pressure liquid chromatography.
So what are the advantages, which seem obvious to me, but is there any other advantage why this would sell into the marketplace?
Well, already as a small laboratory scale instrumentation, which we're starting with, there is a large overseas market for this. As we know, in the scientific market is very large overseas and growing tremendously in China and Japan, and actually scientists in those countries are familiar with this kind of technology, and they really want to undertake using this new method, which is the spiral countercurrent chromatography.
Where was it developed?
This is developed at the National Institutes of Health by a very prolific scientific inventor, Dr. Yoshiro Ito. And my company was established. We have the exclusive license for this design of chromatograph.
How large do you think the market is?
The instrumentation market is tremendous. It's about 42 billion worldwide, analytical instrumentation market. Now, this is a very small part of it, a small niche right now, but it could grow significantly if it is taken on by the industry and the research industry in the United States and Europe, all over the world, and grows into the protein application. So I figure this company could grow to a few million, maybe even 50 million with success in some of these applications.
Question for you is, how much does it cost to manufacture this?
Well, this is a certain amount of cost, but this is made by an advanced manufacturing technique, laser sintering, and they can run off maybe 6 of these after the computer design. So this is not very expensive. Pricing for this right now runs around $3,000 or $4,000, but it can be used and over again. Unlike a chromatography column that is filled with solid support, the purchase of that is about $1,800, but it can only be used for a certain amount of uses. So this is a low-cost method.
At the end of the day, does this product, compared to the competition that is currently used, give you a more refined sample?
It does. It is able to do so, especially for the presently difficult molecules like membrane proteins and non-water-soluble peptides. This has the solvent systems that can handle those compounds and give a good recovery and high purity.
Does it need any approvals?
The instrumentation itself will not need approvals, but to be used in a process for a pharmaceutical company, that whole process will have to probably be approved.
Okay.
But that's not our problem
Got
right
it.
now.
Exactly. What is your website for other people to come and visit with you? And I also, before you do that, what's your background?
Well, I have previous degrees in peptide chemistry from the University of Colorado Health Sciences Center from a famous peptide chemist, John Morrow Stewart, and I received a PhD from Johns Hopkins in biochemistry.
And your website. Wait. You want to add?
Fact,
Go
yes.
ahead.
I'm the former president of Peptide Technologies Corporation. That we were in the peptide industry for 20 years, producing peptides. And my website is ccbiotech.us.
And any special spelling, or is it simple?
It's I believe it's simple, just www.ccbiotech.us.
Ladies and gentlemen, Dr. Martha Knight of CC Biotech LLC. It's a private company, and we are at the William E. Hanna Jr. Innovation Center at Shady Grove in Rockville, Maryland. And I want to thank you for coming on to SNNLive.
Thank you very much.
Good luck to you.
Thank you.